Hello friends, so welcome to lecture 2 of week 6. So in this lecture we will look at the use of link reads and also how to generate it. So before going into link reads and its processing and basically the other kinds of link reads such as the 10x Chromium or the Illumina Tell-Seq link reads, we will see that what is the significance of link reads, and before that why we need to generate this link read. So basically if you see that the first first sequencing of genomes was done by Sanger sequencing that produced long reads, and it was a very cumbersome tedious process where each fragment was transformed and then colonies were picked and then basically it is sequenced. But now it was followed by the second generation sequencing where which includes Illumina also, right? Pyrosequencing or basically sequencing by synthesis, right? And so and ion torrent is also one of the new technology in that. So basically these are producing short reads. So basically the link reads that we are going to see today are short reads which are produced by any of these short read technologies, but the library preparation is done by the 10x Genomics Chromium instrument, right? So basically that is now the third generation sequencing, which includes the long reads such as PacBio or Nanopore ONT, which we have already covered, and the link reads which are actually the short reads which are generated by the Chromium 10x Genomics instrument. So basically we will look at how these are generated, what is the technology behind, and how these reads are then analyzed further, right? So Chromium platform is developed basically with the purpose of doing single cell genomics, and producing link reads is one of the outcome of it. Okay, so basically it is they call it as next gem technology, okay, which for the analysis of single cells and massive scale, including the genomics, transcriptomics, right? And basically it has thousands of single cell partitions with barcode for downstream analysis. So it includes partitions which are well barcoded, so that when the part of DNA or basically cells which goes into these wells or single cell partitions, they are uniquely barcoded, okay. And this can be used for the link read sequencing for genome sequencing. So basically how it works is you will have the gel bead cells or nuclei enzymes, and basically these are loaded into the next gem chip, okay, which looks something like this, right? So this goes into the Chromium instrument. So basically this is on the chip, and it's a fast process also. In few minutes it can produce the libraries. So basically in this you can see that these are the beads which are going through, right? And cells and nuclei and enzymes are pushed from here and attached to those beads. So we will see this process in detail, that how these are attached, but the idea is that it will have beads with unique barcodes, and whatever goes into one bead will be labeled separately and treated separately, so that it can be resolved later and can be considered as a part of the link read for that barcode. Okay, so it may have tens of thousands of gems in per run. Okay, so basically once this single cell gems are ready, okay, the DNA fragments that are done are also given the same barcode like this, you can see. Okay, so there are like 3 different DNA fragments which are given the same barcode, and after that basically this after barcoding, basically all fragments from the same cell or nucleus share this common 10x barcode, okay, so which are coming from same fragment or the same nucleus, right? And this has further basically sequenced using any Illumina instrument, right? So basically the core of this single cell genomics or 10x link read is the single cell gems, right? So which we will see in detail in the next slide. And basically before that single cell genomics can also be used for single cell transcriptomics or single cell gene expression. So where the idea is to capture the 3' mRNA and then look at the expression of those genes, or basically recovering the RNA and also their assembly. Okay, so it works like this. Basically, you will have this is like overall workflow. So you will have the cell or nuclear suspension which goes into the chip, that goes into the instrument, and that produces the sequencing ready library, followed by the sequencing and the data analysis. So we will see this process in detail, and there are two instruments from Chromium. One is hands manual, and basically other is automated, and which provide the support for the single cell genomics, and basically this one is used for the Chromium X for the link read sequencing. Okay, so basically the approach is like that. It's done on a like very small benchtop instrument which is called Chromium, right? And basically molecular barcodes are used to tag reads that come from the same DNA fragment. Okay, so like this you can see in this picture, from this DNA you can, like for example, just for example, 3 pieces are generated, the fragment 1, fragment 2 and fragment 3, which may have some considering it separates 2 genes, some overlapping fragment and like that. Okay, and then each fragment is attached to one separate bead, right? That means the separate barcode also, and then this is further fragmented with the and assigned the same bead, right? And once the sequencing is done, you can resolve this or demultiplex this based on the whitelist of the barcodes, right? So basically the idea is that it can provide long range information which is missing from short read, because it is generating the reads from a long piece of DNA, right, or long fragment of DNA, and that also these are link reads which are producing from 3' and 5', 5', 3' end, right. So this can generate information about, so it can locally assemble that fragment and also can generate long distance assembly information. So each single use chip can process up to 8 samples together, right. So that means 8 different libraries you can load here, and which can be done in less than 20 minutes, so which is fast also, and these libraries then can be used for any Illumina instrument. So Illumina read sequencer basically, right, and then followed by the analysis using Supernova or before that the 10x scripts that we will see. Okay, so basically the DNA fragments are barcoded and sequenced. So this is the basic strategy for this, right, and unique barcode is given for each fragmented molecule, right, which later helps into resolving or separating the different fragments. Okay, so again, like to see this in detail, you can see that there are 3 different fragments which are given the which are bound to the gems and basically given the same barcode, and after the barcoding they are sequenced, and the sequences now contain the information of the barcode also at the 5' end, and basically that can be later be used to resolve this sequences separately and then for generating the larger contigs. Okay, so link reads have been used recently in several genome sequencing, metagenomic sequencing, and also comparing with other technologies like Hi-C, or also for their basic, or one of the important outcome is that it can be used for a diploid genome to resolve the maternal paternal chromosomes, right. So it was developed in 2015 and it was named GemCode or Chromium. Okay, so basically it's a gel bead based barcoding, right, and followed by the sequencing. Later on the link reads V2 version also came, which employed separate barcodes for the beads as well as for the DNA to resolve better. Okay, and since it is a microfluidic instrument, you need very little amount of DNA, about 1 nanogram, whereas another link read technology, which is Tell-Seq, can use up to 0.1 nanogram also. So basically it needs very less amount of DNA, right? And basically that 1 nanogram is spread across about 1 lakh droplets, in which the PCR happens to amplify the DNA fragments for barcoding and followed by the sequencing. Basically this can help you to generate long range information from short reads, and that is why it is called link reads. So it has several applications, for example de novo genome assembly that we will discuss. So it can be used for bacterial assembly, it can be used for eukaryotic assembly. The only limitation is that genomes above 3 billion base pairs or 3 GB in size may be difficult and is not recommended, whereas all the smaller genomes are fine. And also there is a cap on the number of reads and the genome size and the number of reads and the basically coverage of the genome. For example, you cannot go beyond 60x or less than 60x, otherwise it may not work. And while you are doing the genome assembly, you can also do the haplotype phasing, which for example we will also discuss. We can also study the structural variation analysis, because it is giving the long read or basically link read or long distance information. Transcriptome analysis, one example we will also see, and also for the epigenetic analysis. Okay, so basically the technology is like that. There is a bead of DNA, okay, which is barcoded, and that attaches to one fragment of DNA, okay, and that fragment also gets the same code, right? So this is like barcoded amplicons generated in gel beads provide building blocks of the genome. So this is the barcoded amplicon, right? So it starts with the 10x barcoded gel beads, right, which passes through this tube-like structure, and basically each bead is attached with one high molecular weight DNA. The 1 nanogram that we have taken is basically divided into about 1 lakh beads, right, and then the beads are attached to the DNA, right, and then this bead associated DNA are attached, and in between you can see that oil is also inserted, so that the DNA gets encapsulated in oils, right. So basically beads in oil emulsion kind of thing, right, and you can see each of these bead with DNA and inside the oil emulsion, right, followed by the isothermal incubation to basically amplify the fragment, amplify the DNA, and also attach the beads, attach the barcodes to the each of the DNA fragment which was attached to each of these beads. Then the oil is removed and the 10x barcoded amplicons, because you also performed one amplification step for these fragments that were attached to the beads, right. So those are then sequenced like this. So basically read 1 and read 2 are generated, and in between there is a genomic DNA insert, and this can provide the long distance information. So you can see that about 0.5 to 1 nanogram of DNA, which is equal to about 150 copies of the genome, is partitioned into 1 million of these gems, right. So that means you are actually generating DNA fragments. So in detail, like 150 genomes or 150 genomes of the same type, or what you have what was the starting material, is partitioned into 1 million partitions, and each of this gem contains about 1 by 6,000 of the genome, that is something about 500 kb, right, and basically if there are 10 molecules per gem, so basically each molecule or each DNA fragment length will be about 50 kb, right, and the chances that basically 2 molecules that cover a locus are in the same gem is very is like, that means like with this thing it is 1 in 6,000, right, and the percent unique barcodes are in given genomic locus is 99.98. That means basically they are uniquely separated and distributed, right. So the read coverage recommendations for 10x Chromium instrument is like it will need about 60x coverage at the max, right, from basically from like 35 to 60x coverage, and but 55 to 56x coverage is like works best. Also depends upon the genome size, and for the detecting the structural variations about 25x coverage. Okay, and it is not that if you increase the number of reads like you do in case of short reads or long reads, it is going to enhance the assembly quality. It is not the case, because instead you can do multiple of these libraries, right, sequence separately, and then once you have assembled them, you can merge the assemblies later, right, or you can do hybrid assembly with nanopore or only short reads later, right. So basically the smallest genome it has covered is about 78 MB, just for example, from the 10x website, and the largest genome like for example one of the large genome like 12 GB, or this is frog or aloe vera which is 16 GB, is beyond the scope of 10x genomics or linked read sequencing by 10x, because the software also cannot handle, and basically the number of reads that you will be generating given the 55 to 60x coverage of say 12 GB genome will be huge, and that cannot be assembled by the assembler, right. So generally like Supernova can do best for up to 3 GB of the genome, which is the size of human, but actually till that size you cover almost all the mammals and including humans, a lot of plants, which because plants generally range from like for example banyan tree is just 500 MB, people tree is also similar, and turmeric genome is about 1.2 GB, right. Similarly for amla 1.3 GB. So basically most of the genomes and this you can cover. Plants can be little tricky given their repetitive content and heterozygosity, but and basically polyploidy also, but still those have been done successfully by the 10x read sequencing also. Okay, so basically 10x offers a very good link read sequencing technology, which is very good for like for example human, birds, lizards, or like any diploid genome. Bird genome also range like about 1 GB in size, so birds can most of the birds can also be done by this, and maximum genome size it has done is like 7.5 GB, but basically your limit is like 2.14 billion reads. So in that case the X or the coverage of the genome will like reduce greatly if you go to that high genome size. Okay, and many de novo genomes have been generated by linked read sequencing so far, but mostly it was in combination with other sequencing technologies also, because hybrid genome sequencing as well as assembly works best, right. So plants have also been done, but may be risky because of their polyploidy and high heterozygosity. Okay, so overall, like just to summarize, it works as a there are gel beads, okay, in which basically you attach the DNA and insert that into an oil droplet, in which you do the amplification, right, and then fragment the DNA, and basically each fragment that is there is assigned with the same barcode. Okay, so there is a whitelist of barcodes which is compared by the scripts to segregate the fragments later. Okay. Okay, so proc10g basically is a package or series of Python scripts, and mostly like two main scripts that are used to remove the barcodes and primers and these things from the 10x reads, and also to convert the BCL2 FASTQ format to the FASTQ format, because BCL FAST2 cannot be processed. So there are two scripts, which is process 10x read pi and filter 10x read pi. So first one basically identifies the barcode, labels it, also converts from BCL2 to FASTQ, and the second one helps you to resolve or separate on the basis of barcodes. Okay. So the process 10x reads.py process the FASTQ file generated from BCL to FASTQ, right? And then it looks for the gem barcode, that is the first 16 base pair of the read one, right, or basically the read, right? And for both sequence and quality, that means whether the barcode is intact and also whether the quality of that barcode or the ambiguity in the barcode match mismatch with, compared to the whitelist, what is the status for that. And basically also trims the primer from the read, which is the next 7 base pair of the read, right? And then it compares the barcode that it has extracted to a whitelist of barcodes. Okay, so whitelist is a list of all the barcodes. We have seen that like millions of fragments went into sequencing or basically for the library preparation. So now there will be so many barcodes which are there, right? So it will assign the sequences to these barcode sequence, like for each barcode it will assign the number of sequences, that for each barcode how many sequences were there, that tells that what is the coverage per barcode and also like how many sequences or reads were assigned to those barcode. Okay, so and then it will also label that each read with match, mismatch, ambiguous and unknown based on the quality of barcode. If the barcode matches exactly with the database or the whitelist, okay, it is given a match. If there is a mismatch or basically some error or kind of ambiguity in the or mismatch in the barcode, it is called mismatch. If it is ambiguous or basically it cannot resolve, it is ambiguous. And if it did not find a barcode or barcode was unknown, it is called unknown, right? And then this is further processed by the next skip, that is the filter 10x reads py. So this is the details of how you can use, but the command that I have showed in the previous step is the right one, and this can provide like details of what all you can do, like looking for all output, all reads, not just those that is valid gem barcode. That means you will be processing all the reads and not only those which are match tag, right? And can you do you want to trim the barcode or trim additional bases after the barcode, so that you are sure that barcode and basically you are dealing with clean sequence and barcode and something next to this is removed, right? And then -1 for read one file forward and -2 the reverse read file, right? So that script actually produces a list of barcodes and the number of sequences assigned. For example, this is the barcode that was found by a match, and you can also select like whether you want only the match, mismatch also allowed, right? And the number of reads assigned to each barcode. Okay, so the next script, Python script, is the filter 10x reads.py, which takes the processed output barcode.txt file that we saw in the previous slide, and the processed R1 and R2 files that we generated from the last step, and the output folder. Okay, so basically based on the barcode status, that is the match, mismatch, ambiguous or unknown, it can separate or prepare the files, and you can keep the barcodes at like depending upon match, mismatch status or their evidence, or barcodes which are assigned a x number of reads, such as 100 reads or at max these many number of reads, because just to make sure that at least you are selecting all those barcodes which have relevant number of reads, right, and not selecting any ambiguous barcode. Okay, so it can be this proc10g scripts can be downloaded from GitHub, and this is the place. Okay, now just because we will be covering Supernova in the one of the lectures later, so just to give you an overview, basically the 10x generated reads can be assembled using the Supernova sequence assembler, which is also offered by 10x Genomics, and you can fill up a form and download this Supernova assembler. So basically it is a very good assembler to assemble the 10x link reads, and also it resolves the paternal and maternal alleles, which is a typical error in the other assemblers, right. So that is the advantage of link read sequencing and what it also achieves by resolving those, right. So it can create the diploid assemblies, thus separating the paternal and maternal chromosomes over very long distances. So basically it makes an edge and bubble kind of basically edge or basically vertex and the bubble kind of thing, so that this is one paternal allele, this is another paternal, this is a homozygous sequence between the two maternal paternal chromosomes, and this could be the maternal chromosome, this could be the paternal chromosome, and then again followed by an edge which is the homozygous sequence between the chromosomes. So this way it can produce like pseudo hap 1 or pseudo hap 2 kinds of assemblies that we'll cover later, right. So basically this is the mega bubble, and you can see that this yellow or golden color is coming from one paternal or maternal chromosome, and this is coming from another one, and it can resolve this, and this is the edge, or you can say the vertex, which is the sequence that does not vary between the two, right. And this way it can resolve the maternal and paternal chromosomes, and this is and basically this helps in the de novo assembly as well as the resolving of this, and you have the option to go for raw or pseudo hap 1 or pseudo hap 2 kind of output. So because it is basically keeping so much data, right, in memory, so it requires a large amount of RAM, right, and about 500 GB RAM, and basically this is one of the most RAM intensive genomics software that you will come across for the genome assembly part, right. And also appropriate amount of free disk space per GB. For example, your genome size is 3 GB, so you need 1.5 GB of free disk space, right. So like half a GB per 500 GB for per GB. So if your genome is 3 GB, you will need 500 into 3 or basically 1.5 TB of genome of the basically the disk space, and a 16 core or higher processor, and higher the number of processor will be better because you can split the jobs into multiple pieces. Okay, so the paper for Supernova was published here, and this tells that how the library can be prepared and also the later assembly components. Now link reads have been used for assembling large and complex polyploid genomes also, and for example in case of this genome where they were using, like they took the B37 reference genome, right, and you can basically they showed that 91% of link reads were contigs were accurately assembled, which is a good achievement, and basically the having the complex polyploids is challenging, and link reads were supposed to do till the diploid level, but it has also done for the polyploid, and they were successfully able to resolve the two genomes of the recent developed lot, that is the millet, okay, and their assembly covers almost 83% of the 1 GB genome, and consisted of like much lesser number of scaffolds and a very high N50 compared to the previous reference genome assembly. So they showed that how link reads could be used to improve the previous reference assembly, right. Similarly, another paper assessed the human diploid genome assembly using the 10x link reads, and in this basically they could show that the long basically they showed that what should be the optimal amount of DNA to be generated in terms of coverage for the best assembly quality, and also what should be the optimized length, weighted fragment length, which was 5 to 50 kb, 50 to 150 kb, and also the other parameter that you should keep while you are doing the diploid genome assembly for humans, for example. Another paper showed the significance of link read technology in case of metagenome assembly, right. So earlier, like if you see the metagenomes suffer because generally like short reads are used for their assembly, which leads to like smaller quantities or inappropriate assembly, right? Even the draft genomes that you produce by only by short reads still have more than 100 pieces, and which is not appropriate for such small genomes which are only 5 million base pair or something. So basically link reads can be used, okay, and they showed that up to 10 to 100 kb long DNA fragments they could generate, and they also showed the effect of read depth on the metagenome assembly and how the link reads can be used for that. And ultimately, like they also showed that the final assembly that they generated by link reads was not like similar to PacBio long reads, but it was better in the sense that PacBio, since it is very expensive technology, but still by using link reads and like short read technology, which is much cheaper than the PacBio, a reasonable quality assembly, which is high quality as well as low cost, could be achieved. In another case, some authors compared the Hi-C, which is basically used for chromosome level assembly, versus the 10x genomics, right, for the whole genome phasing. So phasing is one of the features of linked read sequencing, and they could found that using the 10x phasing, they could get the best phasing performance among the methods analyzed, that is 10x and Hi-C, and they are the highest phasing rate 89.6% long adjusted N50, and lowest switch error rate, which shows that basically it worked better than the Hi-C in terms of the phasing. Then ARCS is one of the scaffolders that can be used for the link reads data, and basically the author showed that on a ABS assembled Homo sapiens genome, that is a human genome, it can improved the genome assembly by 6 folds, and which showed that basically link reads can be used in like along in place of the short reads assembly, and it provided like 6 times better assembly as compared to only the short reads. Okay, another link reads. So basically we were discussing the link reads by Chromium. So there is one more technology that is Illumina supported, that is a Tell-Seq, and it can also do the link reads and by a different method, completely different from the 10x genomics, and that is also coming up and has been used recently. So basically they produce the Tell-Seq WGS library preparation kit is available, which can produce the link reads by Tell-Seq method, which is a transposase method, and it uses a transposase enzyme to basically construct these link reads, which can then be sequenced by any of the Illumina platforms, including Mini-Seq, MySeq, or NextSeq or NovoSeq also, but iSeq I think cannot be used for this, and then the analysis can be done at base space, right. So the advantage of this one is like even like 0.5 nanogram of input DNA is sufficient for the Tell-Seq technology, right. So it works like this. So basically there is a transposon, a transposase, and a tell bead with the barcode. Okay, so basically the transposome, the bead, and the DNA are taken together, which leads to the barcoded, or basically the DNA is captured by this okay, and then segmented, right, and after it is broken, basically it is amplified and you get the link reads or basically the reads with the inserts, right. So for better clarity, basically there is a DNA fragment, a transposon, transposase, and tell bead. Okay, so basically this transposon and the transposase are assembled together, right, like this, which forms the transposome, right, and which are attached to the bead along with the DNA. Okay, so this is basically now the barcoded DNA fragment complex, okay, which is cut, and basically the DNA is then used for the sequencing. Okay, So they basically the transpose enzyme linked long read sequencing or Tell-Seq generates barcoded link reads for genome scale haplotype phasing and de novo sequencing. So you start with very little amount of DNA which is barcoded and amplified, and then after the quantification and library checks that you do, it can go for sequencing. So this process takes about 3 hours, and the kit is provided by Illumina. Okay, and after that basically this is the insert and reads are generated from both sides, read 1 and read 2, like this. And once you have the read, so they also provide a assembler called Turing assembler for assembling the link reads that are generated by the Tell-Seq WAS library preparation technology. Okay, so just in terms of the time taken in different steps, so basically to barcode DNA takes 15 minutes, to stabilize 30 minutes, segment 10 minutes, then washing the beads 20 minutes, and basically after this you can pause or stop also. Okay, then you amplify the library which takes about 40 minutes, and clean up the library that takes 25 minutes, and then followed by the quantification and quality check which takes about 40 minutes. So in total like 3 hours are sufficient to create the Tell-Seq link reads library, which can then be sequenced. And like for genome size, for example, it is like 1 to 50 MB, like that means it will cover all the bacteria or up to fungi genomes or smaller micro eukaryote. 0.5 nanogram of DNA is sufficient reaction volume and the standard size kit, and basically for 50 to 100 MB 1 nanogram, 100 to 200 MB 1.5. so basically this input amount also increases because you want a sufficient representation of the genome. So that means you need more input DNA so that the library that you generate from this sufficiently covers your genome. And basically sequencing can be done on any Illumina platform except the iSeq, which is like a small, very small instrument and cannot generate too much data. So the difference between Tell-Seq link reads and 10x genomics link reads is that Tell-Seq depends upon transposase to insert the barcodes into the double-stranded DNA, whereas 10x genomics reads basically partition the single-stranded DNA into various beads, and then it labels the barcodes. And Tell-Seq has much higher or larger barcode capacity, more than 2 billion versus 4 million in case of link reads, 10x link reads, and therefore basically it can accommodate even larger genomes also. And Tell-Seq does not require like basically or you can say that Tell-Seq uses all the components which are well supported by Illumina or standardized by Illumina, whereas in case of and basically the library preparation cost is also even half of the 10x genomics reads, and so maybe offering a cheaper platform in terms of the link read genome sequencing. Okay, so basically by link reads we are trying to address the issue of genome complexity that we commonly face, such as the heterozygosity and ploidy, and also the repeats to some extent. But basically for repeats you have to have the long read sequencing data that you generally get from nanopore or PacBio, whereas heterozygosity and polyploidy can be addressed, and for example in heterozygosity in case of maternal paternal allele can be resolved by the 10x Supernova assembler. Okay, and also the ploidy, which is one of the challenges in case of the plant genomes. Okay, so coming to sub example, for example turmeric genome link reads 82x 10x genomic link reads and 41x nanopore reads were generated. So even though the reads were more, while running the Supernova we have to specify how many reads we want to use, right? And basically it led to about 100 kb of N50 and 92% of boost co-completement. That means the genome coverage or the coverage of all the single cluster orthologs was almost 92%, which is reasonable. And also you have to see that it is a highly heterozygous genome with it's a triploid genome. So that also caused challenges into the assembly, right? And when only the 10x genomics reads were used, it led to a genome assembly size or N50 of 15 kb. Along with the long reads, only long reads, it gave the nanopore N50 assembly. N50 only by the long reads gave N50 was 60.9 kb or 61 kb. But when the two were combined, or in case of hybrid assembly, the final N50 achieved was 100 kb. That means combining the two approaches helps by increasing the boost score. For example, only the 10x genomics link read using the Supernova, the boost score was only 75%. In case of nanopore assembly, only nanopore assembly, the boost score was 71.5%, but when we combine the two, it increased the boost score to almost 92.4%, which is a advantage of using a hybrid genome sequencing and assembly technology. And similarly for the Jamun genome, a high amount of 10x genomics link reads and the 20x of nanopore long reads were used, which resulted in a good N50 of 1.179 kb and boost score completeness of almost like 98-99%, which is a very good outcome. So that means that genome coverage was almost complete and you could cover all the single cluster orthologs. So in this lecture we looked at what are the different types of link read sequencing technologies, including the 10x, which is offered by Chromium, and the Tell-Seq link read technology, the ways of cleaning the reads, and also some overview of the assembly, and some paper that discusses the advantage of link reads and in various type of genome sequencing or transcriptomic projects. And at the end we saw a few examples where both the link reads and the long reads were used to assemble genomes, and what was the net N50 and boost score or the genome completeness status after combining the different methodologies. So this is good for today. Thank you.